So what is ciliary body? In our previous video, we talked in detail about the anatomy of the iris. And today, we shall be discussing in detail about the anatomy of the ciliary body. The ciliary body basically is the middle part of the uveal tract and is situated between the iris and the choroid. In this diagram, the choroid or the uveal tract basically has been drawn in brown color, and the orange color basically represents your retina. More specifically, the ciliary body is considered to be the forward continuation of the choroid at the ora serrata. So basically, the retina ends at the ora serrata, and right at that point, the choroid is also ending and becoming the ciliary body. Coming to the shape of the ciliary body is triangular in cross section and it has three sides. It has an anterior surface, an outer side, and an inner side. The anterior side basically takes part in the formation of the angle of the eye, and in the middle of the anterior part, the iris is basically attached. Now, this anterior part can also be called as the anterior surface, or simply we can also call it as the face of the ciliary body. Then we have an outer side. The outer side basically lies in close contact with the sclera that I've drawn over here in grey color, and this is the scleral spur. Now, between the sclera and the ciliary body, there's a potential space which is known as the supraciliary space. Then we have an inner side of the ciliary body over here, and this inner part of the ciliary body can again be divided into two parts. We have the pars plicata, that is the anterior part of the ciliary body on the inner side, and we have the posterior part of the ciliary body on the inner side, which is referred to as the pars plana. Now, let us talk in detail about the pars plicata and the pars plana. The pars plicata, as you can see, it is basically the anterior part of the ciliary body, and this part of the ciliary body on the inner side is thrown into various finger-like projections over here. Okay, and these finger-like projections are called as the ciliary processes. Now this part, this anterior part of the ciliary body which is thrown into these finger-like projections is known as the pars plicata or the pars ciliaris. Whereas the posterior part of the ciliary body is smoother. It does not have all those finger-like projections or the ciliary processes, and therefore this posterior part of the ciliary body is referred to as the pars plana or simply as the pars orbicularis. So as you can see in this picture, the pars plicata is the finger-like projections, and you can see here in this diagram you have these various finger-like projections on the anterior aspect or the aspect of the ciliary body which is more closer to the lens, and that aspect is known as the pars plicata. Then slightly outer to the pars plicata, or you can say posterior to the pars plicata, we have the smoother part of the ciliary body which is referred to as the pars plana. So here this is the pars plana and here this is the pars plana, right? And then more outer to the pars plana is basically your ora serrata, that is the ending of the retina, okay, and that is marked in the orange color in this picture. Now there are some important measurements that we must know about. So first is the pars plicata. The width of the pars plicata is uniform all around. So you can see the central part, the central circle represents the lens, and then we have the anterior part of the ciliary body surrounding the lens, and the width of the pars plicata is about 2 to 2.5 millimeter width, and that is actually uniform all throughout the 360 degrees around the lens. Okay, whereas this is not the same for pars plana. Pars plana, as you can see, it is about 5 millimeter wide temporally, whereas nasally the pars plana is slightly thinner or has slightly less width, and that is about 3 millimeters nasally. So these measurements are quite important. Now here's a clinical nugget. The pars plana is relatively avascular. So what I mean to say is the pars plicata, which has those folds in it, which are called ciliary processes, is basically having more vascular supply compared to the pars plana. And this makes pars plana relatively an avascular structure. Therefore, this route is utilized to access the vitreous cavity. So in cases of pars plana vitrectomy or while giving intravitreal injections, the injections or the instruments can directly be introduced into the vitreous through the pars plana and not through the pars plicata, and this is done to avoid bleeding during the procedures. Now the question is, at what distance the pars plana is located from the limbus, or how does a surgeon basically locates the exact position where they're supposed to make an entry into the vitreous or into the eye? The answer is very simple. Pars plana is basically located at about 4 millimeters distance from the limbus in case of a phakic eye, whereas it is located at a distance of 3.5 millimeters from the limbus in case of pseudophakic or an aphakic eye. Okay, so when the lens is present, the pars plana is slightly posterior, so it is at about 4 millimeters, whereas as the thickness of the IOL, that is intraocular lens in pseudophakia, or when the lens itself is gone in aphakia, the pars plana is situated slightly anteriorly, about 0.5 millimeters, and therefore the distance from the limbus of pars plana in pseudophakic or aphakic eye becomes 3.5 millimeters. Moving on, let's talk about the microscopic anatomy of the ciliary body, which includes the layers of the ciliary body. First, let us try to enumerate the 5 layers of the ciliary body, and then we'll talk about each of these layers in detail. So from outside to inside, we have the supraciliary lamina. First, so let me clarify what is outside. Outside basically means the part of ciliary body which is in close contact with the sclera. So sclera is outside, so this is outside, and the inner side means towards the posterior chamber of the eye, that means towards the aqueous humor and the lens. So from outside to inside, we have the supraciliary lamina, that is totally close to the sclera. Then we have the stroma of the ciliary body. Then we have two epithelium. The outer epithelium is known as the pigmented epithelium, and the inner epithelium, which is closer to the posterior chamber, is the non-pigmented epithelium. And finally, we have the internal limiting membrane of the ciliary body. So now let us see them in the diagram form. So we have the outermost layer close to the sclera, and that is called the supraciliary lamina. Then we have basically the stroma, and the stroma actually has the ciliaris muscle in it. The ciliaris muscle again has 3 parts: circular muscle fibers, longitudinal muscle fibers, radial muscle fibers. Don't worry about them because we are going to talk about them in detail now. Then we have two epithelial layers. The outer one, that is towards the stroma, is the pigmented epithelium, and the inner one is the non-pigmented epithelium. And finally, the innermost layer of the ciliary body is the internal limiting membrane. Now let us talk about each of these layers one by one. So the first one is the supraciliary lamina. Okay, now supraciliary means that it is actually sitting on top of the ciliary body, right? It literally means that. Now it is basically formed by the outermost condensed part of the stroma of the ciliary body. Now something similar we discussed when we talked about the iris, and that was the anterior limiting membrane of the iris. So it is usually considered that the supraciliary lamina, anteriorly it continues with the anterior limiting membrane of the iris, and posteriorly it is continuous with the suprachoroidal lamina. So it's quite easy to remember. So you have some lamina sitting on top of the uveal tract or on the outer side of the uveal tract. So in the iris, it is the anterior limiting membrane of the iris. In the ciliary body, it is a supraciliary lamina, and in the choroid, it is the suprachoroidal lamina. Coming to the stroma of the ciliary body. The stroma of the ciliary body is basically made up of the connective tissue and the collagen fibers, and mostly it is the ciliary body muscle, which is referred to as the ciliaris muscle, which is present in the stroma. It forms the bulk of the ciliary body stroma. Of course, apart from that, you also have some nerves, you have some vessels, some cells. Particularly, we have the pigmented cells and other important cells in the stroma of the ciliary body. But first, we are going to talk about something which forms a major bulk of the ciliary body stroma, and that is the ciliaris muscle. The ciliaris muscle is a non-striated or an involuntary muscle of the ciliary body. That means that you cannot control your ciliaris muscle. So it is not under your control. It's not a voluntary muscle. It's an involuntary muscle of the ciliary body. Now, just like the ciliary body, the ciliaris muscle is also triangular in shape. And in fact, it has 3 different types of muscle fibers which are divided based on their orientation in the ciliary body. So we have the longitudinal muscle fibers, also referred to as the meridional muscle fibers. Now these are the fibers which are actually attached to the scleral spur. So what is present just outside the ciliary body? It is the sclera, and anteriorly it is the scleral spur. Now, the longitudinal muscle fibers basically are attached to the scleral spur, and they are arranged in such a way that they are parallel to the sclera. So these are your longitudinal or meridional muscle fibers. Then we have the circular muscle fibers. The circular muscle fibers are the anterior-most fibers in the ciliary body, and they are arranged in a circular band-like fashion in 360 degrees around the eye in the ciliary body. Okay, so just like we have the sphincter pupillae, the sphincter pupillae orientation is also a 360 degrees band-like muscle around the pupil. Similarly, here also something similar. We have a circular muscle in the ciliary body, circular muscle fibers of the ciliary body. Then we have the radial muscle fibers, which are also referred to as the oblique muscle fibers, and they are oriented radially in the ciliary body, as you can see over here. Now, it is very important to discuss the actions of the ciliary muscle. Okay, so basically what happens is that we know that the ciliary muscle basically is present in the ciliary body, and from the ciliary body some important structures actually take origin, and those are called the zonules of Zinn. Now these zonules of Zinn are attached to the equator of the lens, and they play a very important role in the accommodation. Okay, so what happens is that normally the normal tone of the ciliary muscle around the lens actually keep these zonules very tight, and it keeps the lens in check. However, when the ciliary muscle is going to contract, okay, it contracts toward the center. So what happens, it moves circumferentially towards or closer to the lens. As such, what happens, the ciliary zonules will actually now become more slackened, or they will get relaxed. And now as the zonules become relaxed, the lens can now do whatever it wants. So it chooses to increase the anterior curvature. It basically increases the anterior curvature of the lens, and we know that when the anterior curvature of the lens increases, the power of the lens increases, and the person can actually see close objects clearly, and that is what is known as accommodation. So if you don't understand this clearly, we have a video on accommodation, and we also have a video on anatomy of lens in which I explain to you in detail about the zonules and the anatomy of the zonules. So coming to the ciliary muscle. So ciliary muscle has a very important role in accommodation, and in particular it is these circular muscle fibers which help in accommodation. Apart from that, remember I told you that we have longitudinal muscle fibers also. So these longitudinal muscle fibers are attached to the scleral spur, and because of the attachment to the scleral spur, whenever there's a contraction of the ciliary body, what happens is that the scleral spur is going to be pushed backwards, okay, towards the ciliary body, and that is going to cause opening up of the trabecular meshwork, a very important point. So in our video on the anatomy of the anterior chamber and anatomy of the angle of the eye, I explained to you what is a scleral spur, what is a scleral sulcus, and what is trabecular meshwork. So if you would remember from that video, the sclera has this groove over here which is referred to as the scleral sulcus, and bridging that scleral sulcus we have this meshwork which is called the trabecular meshwork. So that trabecular meshwork is very important for the drainage of the aqueous. Now, the longitudinal muscle fibers which are attached to the scleral spur, as they contract, they pull the scleral spur backwards, opening up the trabecular meshwork, and they basically help this way in the drainage of the aqueous humor. So now I told you that there are two actions of the ciliary muscle. One is accommodation and the second is drainage of the aqueous humor. Coming to the nerve component of the ciliary stroma, so we have nerve supply of the ciliary muscle. The ciliary muscle basically are supplied by the parasympathetic innervation, and this parasympathetic innervation basically comes from the third nerve. The third nerve basically sends fiber to the ciliary ganglion, and these fibers which are reaching the ciliary ganglions are known as the preganglionic fibers. Now, these preganglionic fibers are going to relay in the ciliary ganglion, and they're going to come out from the ciliary ganglion as various short ciliary nerves. And it is these short ciliary nerves which are going to go and supply the ciliary muscle. Okay, so the nerve supply of the ciliary muscle is basically from the parasympathetic innervation of the third nerve via the short ciliary nerve. Now, at this point, I would like to tell you one more important clinical nugget. The short ciliary nerves, it not just supplies the ciliary muscle, it also supplies the sphincter pupillae muscle, which I explained to you in detail in the previous video on the anatomy of the iris. What about the vessels? So remember I told you that the ciliary stroma is made up of the ciliary muscle, the vessels, the nerves and the cells. So we discussed the ciliary muscle, we discussed about the nerve supply, and now we're talking about the vascular stroma of the ciliary body. So basically the vessels which are present in the ciliary body, they basically form what is called a major arterial circle. And to be more precise, the vascular stroma of the ciliary body consists of the major arterial circle. And this major arterial circle is present somewhere near your circular muscle fibers, and to be more precise, just anterior to these circular muscle fibers in the ciliary body. Now, how is this major arterial circle formed? It is basically formed by the long posterior ciliary arteries and the anterior ciliary arteries. Now, if you would have visited my video on the anatomy of extraocular muscle, you will know that the anterior ciliary arteries are associated with the blood supply of the extraocular muscles. And by the end of this lecture series on anatomy of uveal tract, where I also talk about the blood supply of the uveal tract, you will know what is a long posterior ciliary artery. Okay, so these long posterior ciliary arteries and the anterior ciliary arteries, they join together and they form a circle around the iris, which is called the major arteriolar circle or the major arterial circle. Okay, and to be more precise, this is located just at the junction of the ciliary body and the root of the iris, just ahead of the circular muscle fibers, as you can see over here. Okay, and it is this major arterial circle which is going to actually send the branches, radial vessels into the iris, which are actually responsible for the formation of the radial streaks and furrows in the iris, which I discussed in the previous video on the anatomy of iris. Moving on, we discussed about the supraciliary lamina, we discussed about the ciliary stroma and the structures which are present in the ciliary stroma, and now we shall move towards the epithelium of the ciliary body. The first one is the layer of pigmented epithelium. The layer of pigmented epithelium is also known as the outer epithelium because it is more close to the sclera than to the posterior chamber. Now, if you carefully observe, it is considered to be a forward continuation of the retinal pigment epithelium. Now, if you would remember from the anatomy of the retina basically has 10 layers. However, embryologically, the retina is basically having two parts. The outer part is the retinal pigment epithelium, and the inner 9 layers of the retina together form what is called as the neurosensory retina. Now, since the pigmented epithelium is also the outer epithelium of the ciliary body, it would not be wrong to consider it to be the forward continuation of the retinal pigment epithelium of the retina. Now, similarly, we know that the anterior epithelium of the iris is also the outer epithelium of the iris, that is closer towards the cornea than the posterior pigmented epithelium, and therefore we can also consider that the pigmented epithelium of the ciliary body basically continues anteriorly with the anterior epithelium of the iris. So, to summarize, the retinal pigment epithelium of the retina, the pigmented epithelium of the ciliary body, and the anterior epithelium of the iris are basically the outer epithelia, and therefore they are all correlated with each other. Then moving on to the layer of non-pigmented epithelium, which is also known as the inner layer of the epithelium of the ciliary body. Now this is known as inner layer because it is in close proximity with the posterior chamber which holds the aqueous humor. Right? So here the non-pigmented epithelium has been drawn in lighter shades of the brown color. Now, the inner layer of the retina is the neurosensory retina. So it would not be wrong to say that the non-pigmented epithelium of the ciliary body basically is a forward continuation of the neurosensory retina. And similarly, the non-pigmented epithelium of the ciliary body basically continues anteriorly with the posterior pigment epithelium of the iris, which is basically the inner layer or the posterior layer of the iris. Okay, so we can say that the neurosensory retina is basically continuing with the non-pigmented epithelium of the ciliary body, and that becomes continuous with the posterior pigmented epithelium of the iris. Now, all this correlation basically happens because of their common embryological origin. Now coming to the innermost layer of the ciliary body, and that is the internal limiting membrane. So just like we have the internal limiting membrane in the retina, which is the innermost part of the retina, we have this innermost part of the ciliary body, which is the internal limiting membrane of the retina, which is in close approximation to your posterior chamber. So let us summarize: the anterior epithelium of the iris, the pigmented layer of the ciliary body, also referred to as the outer layer of the ciliary body, and the retinal pigment epithelium basically correspond to each other. At the same time, the posterior pigment epithelium of the iris corresponds to the non-pigmented epithelium or the inner layer of the ciliary body, and that corresponds to the neurosensory retina, which is the inner part of the retina. A very important high yield point that I want all of you to understand and take home from this video. So now moving ahead, let us discuss the microscopic anatomy of the ciliary process. Okay, so we know that pars plicata basically has those ciliary processes, and it is very important that you actually go into the microscopic details of the ciliary processes. So these ciliary processes are nothing but they are actually the finger-like projections coming from the pars plicata, and usually you have around 70 to 80 ciliary processes in eye, and each of them is about 2 millimeters in length. Coming to their ultrastructure, each ciliary process basically is composed of a capillary network, the ciliary stroma, and the two layers of epithelium. Definitely whatever was present in the ciliary body will also be covering the ciliary processes. But now we are going to deep dive into the ultrastructure of those epithelium. Okay, so here also you have the pigmented and the non-pigmented epithelium. So basically, what all are we going to study? We are going to study about the capillary network that you can see in that ciliary process, which consists of an arteriole and a venule. Then you have the stroma, and then covering that stroma, you have the pigmented epithelium. And then the innermost one is the non-pigmented epithelium. And then finally, we have the internal limiting membrane. Now, if you observe this diagram in isolation, it is quite easy to get confused and call non-pigmented epithelium as the outer epithelium. But remember, the inner and outer is not described as according to this diagram. The inner and outer, when we use it for the ciliary body, we use it in terms of the sclera. So the epithelium which is closer to the sclera, which will be the pigmented epithelium, that would be called the outer epithelium, and the epithelium which is closer to the posterior chamber, okay, will be called the inner epithelium, and that is the non-pigmented epithelium drawn in pink color here. Again, very important concept that I want you to remember. Now let's discuss about these parts. So we have a network of capillaries within each of these ciliary process. So each process consists of this network of capillaries. Now this actually consists of an arteriole. This arteriole is actually coming from the major arteriolar circle that I told you, or the arterial circle. So the arteriole basically enters into the ciliary process. It forms this capillary network here, and finally it's going to end in the venule. Now, what is important for you to remember is that these capillary networks, they basically have very thin endothelium, and they have lots and lots of fenestrations or openings which are actually covered by an external diaphragm. Okay, so this makes it a very important site of increased permeability. So because of those fenestrations which are present, what happens is that we actually see a process of ultrafiltration, and what filters out water and ions into the adjacent stroma. Okay, so as you can see over here, because of that excessive fenestrations or pores which are present in the thin endothelium of the capillary network, excessive amount of water, ions are going to diffuse freely from the capillary network into the stroma of the ciliary process. A very important point that you must remember. Obviously, in the next video, or maybe the next, we are going to discuss about the dynamics of the aqueous humor, in which we will discuss how this ultrafiltrate is actually formed. Okay, now One more point to remember is that it is not just it not just allows the ultrafiltration of water and ions, but also allows much more larger molecules like plasma derived proteins. So your myoglobulin, gamma globulins, they can all enter the stroma just because of these fenestration. Now, because they enter the stroma, because such larger molecules and solutes are actually entering the stroma of the ciliary body, what is going to happen is that the oncotic pressure within the ciliary process stroma is going to increase, and whenever the oncotic pressure increases, the water is going to flow to that area, and that is going to lead to ultrafiltration of water, and ultimately we'll have aqueous humor secretion. Now, regarding the stroma of the ciliary process, which is the part situated just outside your capillary network, okay, so this part is called the stroma. The stroma of the ciliary process is very thin compared to the stroma of the ciliary body. Obviously, you have a big ciliary body and then you have small 70 to 80 ciliary processes. So definitely the stroma that comes inside the ciliary process is going to be very, very small. Right now, this ciliary stroma basically separates the network of the vessel that we studied just now from the epithelial layers that are present. Okay, and the stroma again consists of collagen fibers and the ground substances. And ground substances could be the solute of plasma that just got filtered out or that just flowed freely from the fenestrations of the capillary network. Along with that, some protein and mucopolysaccharides as well. Now, moving on to the ciliary epithelium. Now, before we jump into the ciliary epithelium, that is the pigmented and the non-pigmented epithelium of the ciliary processes, just like the epithelium of the ciliary body, it is very important that you understand some of the salient features of the ciliary epithelium. And this actually forms the essence of this video. So, first thing is that the ciliary epithelium is actually a bilayered epithelium because we have a pigmented and we have a non-pigmented epithelium. Again, to reiterate and to remind you, pigmented epithelium is the outer epithelium and the non-pigmented epithelium is the inner epithelium of the ciliary body. Number two, they are neuroectodermal in origin. That means they take origin from the anterior ends of the optic cup. Then they are basically the cells which are involved in energy dependent transport process. And the two layers, although they are different anatomically, they are going to act together as a syncytium. So first let us discuss about this bilayered epithelium. Okay, so again we have a pigmented epithelium, but now here let us go deeper into that epithelium. The epithelium is pigmented, it is an outer epithelium and it is formed of the cuboidal cells. Now because it is pigmented, it has numerous cytoplasmic melanin granules within it. Now, a very important point to remember is that it is actually separated from the ciliary stroma by its basement membrane, and the basement membrane of this pigmented epithelium is basically atypical. Okay, and why is that so? Because it is actually a continuation of the Bruch's membrane. Now when we discuss about the anatomy of choroid, I will tell you what is Bruch's membrane, but you should know at this point that the basement membrane, which is separating the ciliary stroma and the pigmented epithelial layer, that is the basement membrane of the pigmented epithelial layer, is atypical. Okay. Next, we have the inner epithelial layer, which is a non-pigmented epithelial layer. Now, these cells are columnar cells. Okay, the pigmented epithelial cells were the cuboidal cells, whereas a non-pigmented epithelial layer is actually columnar, and they have, instead of having a lot of melanin granules, here you have more amount of cytoplasmic mitochondria and endoplasmic reticulum. Now, these are actually again separated from the aqueous by a basement membrane, but this basement membrane of the non-pigmented epithelial layer is a normal basement membrane. Apart from that, they have very nice tight junctions on the apical surfaces, and this is going to form the blood aqueous barrier. So each of these points is very, very important, and I'm actually going to elaborate on all of these. So it's columnar. Okay, remember that. Then it has cytoplasmic mitochondria and endoplasmic reticulum. It is separated from the aqueous by the basement membrane, and it has tight junctions. Okay, so it's very important that, and also you might have observed that the pigmented layer is actually separated from the ciliary stroma by a basement membrane, And the non-pigmented layer is separated from the posterior chamber or the aqueous again by a basement membrane. So it seems like the two layers are actually opposed to each other with the basement membrane on the either side. And this is called an apex to apex configuration. Now, this diagram actually explains the apex to apex arrangement of the ciliary epithelium. So you can see here we have the fenestrated capillaries, which is basically indicative of the capillary network that you see in the ciliary stroma. So this side is actually your ciliary stroma, and then you have the aqueous in the posterior chamber on the other side, on the top of the screen, right? So on the top you have the non-pigmented epithelium, and on the bottom you have the pigmented epithelium drawn in brown color. As you can see, the pigmented epithelium basically has these pigmented cells, or these are called the melanosome, the melanocytes with the melanosome. Those are the pigmented granules within the melanocytes. Whereas the non-pigmented epithelium does not have these melanocytes. Instead, what it has is the mitochondria, and along with mitochondria, it will also have the smooth endoplasmic reticulum. Very important. And if you would carefully observe, on the either side of these cells, we have the basement membrane. So again, here drawn in purple is the basement membrane of the non-pigmented epithelium, which is separating the non-pigmented epithelium from the aqueous humor in the posterior chamber. Whereas on the outer side of the pigmented epithelium, you have again a basement membrane. And remember, this basement membrane is atypical, and because it is actually not a true basement membrane but a continuation of the Bruch's membrane, and it is separating the pigmented epithelium from the ciliary stroma. Now, I will talk more in detail about these gap junctions, tight junctions just in a while. So now, I told you that these epithelial layers are basically neuroectodermal in origin. That means they take their origin from the anterior ends of the optic cup. Now, if you would actually remember from the embryology videos, basically what happens is you have this optic vesicle, and the lens basically then is going to form, is going to invaginate. The lens placard is going to invaginate within that optic vesicle, forming an optic cup. So the optic cup also has an outer part and an inner part, right? So what happens is that the anterior most part of this optic cup basically is going to form your iris and the ciliary body. And since this optic cup basically is formed from the neuroectodermal tissue, we say that embryologically the iris and the ciliary body are also formed. The epithelium of the iris and the ciliary body are also neuroectodermal in origin. So that is one important point you must remember. Now, what about energy dependent transport processes? So basically these cells actually have characteristic of any cell that would be involved in the energy dependent transport process. So similar cell is the epithelium of the lens. Okay, so there also we have more number of mitochondria, more endoplasmic reticulum, and energy dependent means we also need some sodium potassium ATPase pumps. Okay, so all these are present within the cells. Okay, so in these two epithelial cells we have mitochondria, we have endoplasmic reticulum, and we also have sodium potassium ATPases. Along with that, we also see the presence of carbonic anhydrase enzyme, and all these actually give it the characteristic of a cell that is involved in energy dependent transport. Okay, more about this energy dependent transport in the video on aqueous dynamics. Now, what did I tell you? That these two layers basically act as a syncytium. So, although these two layers are anatomically different, that means we have a pigmented layer and we have an inner non-pigmented layer represented in the pink color. Okay, but still, because of the presence of gap junctions, so what are gap junctions? I'll tell you. But because of the gap junctions, they are going to behave as if they are one entity. That means they are going to behave as a syncytium, right? So now let us discuss about some important cellular adhesion molecules, right? So first one is the gap junction. So here, as you can see, basically these gap junctions are these low resistance channels which are present between the non-pigmented and the pigmented epithelial cells, And these channels are actually, they facilitate easy transport, easy diffusions of the ions and other molecules from one cell to another. So basically they are helping in electrical coupling of these two cells, that is the pigmented and the non-pigmented. And because of that easy electrical coupling, because of that easy pathway for the flow of the ions between the pigmented and the non-pigmented epithelium, these two cells are actually going to act together as a functional syncytium, right? Now, why is it so important? What is the importance of this ion transport through the gap junction? So, ion transport between the epithelial layers, it appears to be critically required for the secretion of the aqueous humor. In a research, what they did was they actually inactivated this molecule which is present which is very important for the formation of gap junctions, which is called connexin. So once they inactivated connexin, they found that the gap junctions did not form between the epithelial layers, and this actually led to a substantial reduction in the aqueous humor production as well. So they came with the hypothesis that the presence of gap junction is really important and it's critically required for the secretion of aqueous humor as well. Now, what about this another molecule that you see in this blue color on the side of the pigmented epithelium? That is basically a desmosome. So we have two other types of connections which are called the adherents and the desmosome. So they are basically responsible for giving a structural strength or structural support between the various cells of the pigment and the non-pigmented epithelium. So gap junctions basically connect your pigmented and non-pigmented epithelium, whereas the adherents and the desmosomes are going to connect individual cells to each other. So two pigmented cells or two non-pigmented cells. Now, what about the tight junctions? Now, tight junctions are basically present on the apical surface of the non-pigmented epithelium of the ciliary body. Okay, so they are called zonular occludente, and they're basically thought to be very important component of blood aqueous barrier. Now, these tight junctions, they usually do not allow the passage of any solute through them, and that is the reason why they actually maintain a nice functional barrier between the ciliary body stroma and between the aqueous humor which is present within the posterior chamber. Now, according to a new hypothesis, what has been seen is that according to a new paradigm, what is believed is that we know that the plasma proteins basically are going to actually leak from the capillaries which have high fenestrations within the ciliary process, right? So these plasma proteins are going to leak into the ciliary stroma because of the fenestrations present in the capillaries of the ciliary processes. However, they cannot pass directly into the posterior chamber of the eye because the tight junctions which are present in the non-pigmented epithelium of the ciliary body, right? So you have this high concentration, about 74% present in the ciliary body. Why? Because it's coming from that free passage of the proteins from the capillaries. However, because of the presence of the blood aqueous barrier formed by the tight junctions between the non-pigmented epithelium, it's not able to pass through the posterior chamber, and therefore the percentage of the proteins in the posterior chamber is almost negligible. Whereas, however, what can happen is that these plasma proteins can actually diffuse into the iris stroma, traveling through the stroma of the iris root, and then from there it can freely pass into the aqueous humor or into the anterior chamber, because you know that the anterior surface of the iris is not covered by an epithelium. So we just have an anterior limiting membrane on the anterior surface of the iris, which is basically the condensation of the stroma. And since we do not have an epithelium on the anterior surface of the iris, that protein can actually diffuse into the anterior chamber of the eye, into the aqueous humor through the iris. So that's a new concept about the blood aqueous barrier, which says that the posterior chamber is totally free of the proteins because of the tight junctions present within the non-pigmented epithelium. However, some amount of protein can actually diffuse into the iris through the root of the iris, and from the iris it can easily diffuse into the anterior chamber. So here's the clinical nugget. What forms your blood aqueous barrier? So obviously the tight junctions between the cells of the non-pigmented epithelium, I already mentioned. Apart from that, even the tight junctions are present between the posterior iris pigment epithelium. So this posterior iris pigment epithelium also does not allow the proteins which have diffused into the iris, and they cannot diffuse back into the posterior chamber because of the presence of this tight junctions within the posterior iris pigment epithelium. And apart from that, even the iris vessels are, they do not have any fenestration. So three structures that form blood aqueous barrier are the tight junctions between the cells of the non-pigmented epithelium, the tight junctions between the posterior iris pigment epithelium, and the non-fenestrated iris vessels. Coming to the last clinical nugget of this lecture is the cyclitis. So what is cyclitis? Cyclitis is basically an inflammation of the ciliary body. And whenever there's inflammation of the ciliary body, what happens is that the nerve endings which are present within the ciliary body, those parasympathetic nerve endings from the short ciliary nerve, they get irritated because of the release of the inflammatory products. And this will cause a spasm of the ciliary muscle, and this spasm of ciliary muscle basically causes extensive pain that you see in case of cyclitis. Now, since we know that the ciliary muscle is supplied by the parasympathetic nerve supply, and the parasympathetic nerve supply acts through the muscarinic receptors which are present in the ciliary body. So, in order to manage or tackle cyclitis, we need cycloplegic agents. And the cycloplegic agents are basically muscarinic receptor antagonists. And examples are atropine, cyclopentolate, homatropine, so on and so forth. So that's all for today. I hope you enjoyed it. Thank you and have a nice day.